I'm happy to introduce Bill Johnston, who has come up from San Diego, where he is the public relations director for the San Diego Chargers, and he also serves on the board of directors for the San Diego chapter of the Huntington's Disease Society of America. Huntington's disease is a huge interest on this campus in many areas. You heard about some of the work with Matt and Leslie Thompson this morning. We've got an HD clinical program here as well, so I'm very curious to hear Ms. Thompson.
Thank you. Hopefully everybody got something to eat. I know I did. So if you see food in my teeth, please say something to me, okay? I appreciate friends. I know you would do that for me. I think my job here is to tell the story of maybe a typical Huntington's disease family. And hopefully our story, and I believe our story will be and is typical. My wife Ramona was diagnosed in 1999. Prior to that day, she and I had never heard of this disease. Well, as you probably know, Huntington's is genetic, so how could we have not known? Well, we still don't know. Her parents don't have it, but she does. So it was a shock to us to learn about a disease that is fatal and can be passed on to your children. We have 2 kids. I remember the day that we learned about this disease. We learned that she was positive. Came home, and I just remember standing in the middle of the street in front of our house, crying with my wife. And she was crying not for herself, but she was crying for our kids, who were 14 and 11 at the time. How do you look your children in the eye and tell them that they have a mother who has a fatal brain disease and that they have a 50% chance of inheriting that disease? How do you do that with your children? That was the beginning of a new life for us. It was, the 2 people that you love the most in this world, if you're a parent, you know what I'm talking about, your children. When they learn of something like this, you don't know how they're going to take it. Our son was 14 at the time when he was when he was told of this disease in our family. And he went from a straight A student athlete to doing anything he could to avoid what is now reality in his life. He went from drinking to drugs to barely graduating from high school, to the point where he was living, and he told this to me. He said, "Dad, I'm living like I have the disease now, so I need to know if I have it. I need to find out, because it's killing me now anyway." So when he turned 18, he took the test. He didn't tell me, he didn't tell his mom. He took this blood test and then came to me one day and said, "They have the result and they want you to come with me to learn the result of this test." Well, we learned that he was negative. God blessed us with a negative result for our son. And it's still taken him a while to get through this, but he's doing fine. Our daughter was 11 at the time, and to tell you how kids can take things and deal with them differently, she's now 22. And she has thrown herself into the effort to find the cure for this thing. She's taken the opposite approach. She has gone proactive. She is helping raise money. She's helping bring awareness to what we're doing. So 2 different, 2 kids, but she still also doesn't know. She knows she's going to have to be tested. She doesn't know when. She knows if she wants to have a family, she knows she wants to have kids, get married, she's going to have to know the result of that test. She knows that awaits her. My wife is now living in a skilled nursing home. She was a summa cum laude graduate, elementary school teacher, and now she can no longer talk or walk. Chewing and swallowing is hazardous. She's lost much or most of her cognitive abilities. She still does recognize us when we see her every day, but this disease has taken her away from our home and away from her children. What I wanted to do is I'm going to show something here from 5 young ladies that are affected by this disease who can probably tell the story a little better than I can, how they are personally affected.
My parents sat my brother and I down when I was about 11 and told us that my mom had Huntington's disease.
I remember I had just gotten back from dinner with my family and I fell off of the back of my father's car playing with my sister, and my mother tried to help me because I was crying and my knee was bleeding and she couldn't. She had forgotten what to do and how to take care of me.
I think I was 10 years old and I just didn't really understand what was going on because I didn't really recognize that my dad was sick. And they tried to tell me about what Huntington's disease was and that he was gonna start changing and not really acting like himself anymore.
So far, my mom suffers from a lot of choking problems, like difficulty eating, short-term memory loss has been really big lately, and like fine motor skills, she basically can't write anymore.
My dad that raised us, he's changed. He's not the same person. He's gotten angry sometimes. One day he's fine, the next day he's not slurring his words at all. But it's just not that same dad that was, extremely encouraging at all of our volleyball matches, wanting to do family dinner all the time. It just slowly, it's just watching him digress into somebody that we really don't know.
I think with Huntington's disease, it changes you entirely, even if you don't want it to. I've tried living my life in the ways that I would want to live my life if Huntington's disease never had entered my family. But because I'm always thinking about that, I know it's subconsciously or consciously changing how I've made my decisions, whether I should have gone to graduate school or not, or moved to San Diego away from my family, or just falling in love and wanting to get married.
I felt like having kids is the #1 thing in my life. That's always been, my mom's always said, you've been put on this earth to be a mama. And so for me, I felt like, is my life over? Do I want to find out if I guess in a little bit of a dramatic sense, is my life over?
The only thing I fear is just being alone, Not having somebody, because I wouldn't want to put anybody through what I've gone through. So I couldn't expect anybody to love me like my father loved my mother.
I always thought that once I turned 18, I'd want to go out and get tested right away and know everything so I could plan. But then went to the counselor, turned 18, and then my birthday came and passed, and I still don't know.
I've decided not to get tested at this moment, though there's been times where I just want to get up and drive to the closest place and find out immediately.
What gives me hope is the involvement that my family has within the HD community and how many people that are slowly learning about the disease.
I think awareness is the most important thing. We need to get our voice out there and get our message heard.
We are taking steps towards a cure. I don't know if it'll happen in my generation, in my lifetime, but we are so close that it's possible for maybe my children's generation.
What this does to people and their families and stuff, I just know that there's gotta be a close into this.
I hope that not just for myself, but for everyone impacted by HD, that we find a cure. And if not find a cure, if that's not possible, just to help prolong the symptoms from starting, finding new medications to help with the symptoms so that people who have it can live their lives as long as they possibly can the way they want to.
All 5 of those young women have HD in their family. 4 are at risk, and 1 has tested positive. 1 was my daughter in that video. When stem cell research and the stem cell initiative became top of mind, at least for me, that was just one of the opportunities that keeps us in this world, in our HD world, keeps our hopes alive. To me, stem cell research it's an untapped ocean. It's an endless possibilities, and it's up to people like us in this room and the smart people in this room to make those possibilities a reality. I get to introduce one of those very smart people. Dr. Virginia Mattis is a postdoctoral researcher in Clive Svensson's laboratory at the new Regenerative Medicine Institute at Cedar-Sinai Medical Center in Los Angeles. Currently, she's studying induced pluripotent stem cells generated from several patients with HD in collaboration with a consortium of top HD researchers from 8 labs across the country. Dr. Mattis.
Hello, and thank you so much for inviting me here today, and thank you for all the patient advocates who have shared their stories. You're really why we go to work every day. So as it was mentioned, I am at the new Regenerative Medicine Institute at Cedar-Sinai, just up in LA. I'm in the lab of Clive Svensson, and it was actually due in a large part to CIRM why we're even in LA. Originally we were at the University of Wisconsin, and CIRM made it so attractive to move out to California. The weather's not a bad bonus. So today I'm going to talk to you about using stem cells to model neurodegenerative diseases. We work on several neurodegenerative diseases that I'm going to highlight in this talk. In particular, we've really been focusing lately on Huntington's disease and spinal muscular atrophy. So, excuse me. So why are we using stem cells? Why are we all in this room today? Why is CIRM invented? So in our laboratory in particular, you've heard about this earlier today, described quite well. We're looking for disease in a dish. So what do I mean by disease in a dish? The main things we want to, 1, use cells that have the genes of the disease. Preferably, we want to be able to get cells from a patient. Now, we also want to be able to grow the types of cells that die in disease. For example, in Lou Gehrig's disease, not all of the cells of the body are affected. It's just the cells of the brain that control motor symptoms. Now, with these neurodegenerative diseases, you can't exactly go in and take the brain cells that die out of the patients like you could with someone with a muscular disorder. So we have to use stem cells to grow these in a dish. What we want to do is we want to use those particular types of cells that die in these patients to mimic disease as closely as possible to how it happens in the human. Now, why do we want this disease in a dish? Our goal is to be able to study why certain types of cells die, why the brain cells die, while other cells are perfectly healthy. And we want to use these types of cells that are quote-unquote susceptible to disease to test for new therapies for these terrible, terrible diseases. So, stem cells, and we've all talked about it a lot today. I'm just going to go through this really briefly. Usually, to harvest embryonic stem cells, scientists will take a fertilized egg and take these stem cells, which in science jargon, we like to say pluripotent. What does that mean? Pluripotent means it's able to make any type of tissue in the body, whether it's brain cells, muscle cells, blood cells, you name it. So in a lot of cases in IVF clinics, a lot of these fertilized embryos will be screened for genetic diseases that are known in the family or such chromosomal abnormalities such as Down syndrome. The fertilized embryos that carry these diseases sometimes can be studied in labs, such as in our lab. For example, we have studied embryonic stem cells that have Down syndrome. Now, as I'm sure most of you know, Down syndrome arises when instead of 2 copies of chromosome 21, you have 3. This is the most common chromosomal condition. I'm sure everyone in the room is familiar with the syndrome. And people with Down syndrome have impaired cognitive abilities. And in terms of the science of this disease, what's important to note is that people with Down syndrome often have smaller brains and there are less brain cells there. So we wanted to see, can we use these ES cells that, as you can see in this picture here, this is actually a picture of all the genes. Isn't it beautiful? And you can see that the cells with Down syndrome have 3 copies of this one chromosome, whereas all the other chromosomes have 2. So we wanted to make a disease in a dish for Down syndrome. So we were able to grow brain cells from the stem cells. And you can see here, here's a picture of brain cells we grew in green from a quote-unquote normal person who did not have disease. But look, when we took the Down syndrome cells, they grew less neurons. And this perfectly mimicked disease. We had less brain cells from the Downs. Therefore, we had our disease in a dish. Another way to go about this is because you can't get embryonic stem cells for every disease type, is you can actually take embryonic stem cells and put the disease-causing gene into a virus. Then you can use the virus to infect the stem cells. And therefore, all of the other types of cells you can make, such as brain cells, will also have this same gene. This is another way that we do what we call disease modeling in the lab. So we've done this in our lab also for Parkinson's disease, which you heard beautifully described earlier today. It's a very devastating disease. The neurons that are in control of the movement slowly die, and this results in uncontrolled shaking and difficulty with movement and coordination. So we were actually able to take virus encoding a gene that gives Parkinson's disease and infect our stem cells. We then were able to mature these stem cells into the particular types of brain cells that die in Parkinson's patients. And what you can see from these left 2 pictures, the brain cells in green, these are the exact type of brain cells that die in Parkinson's patients. This is in a quote-unquote normal patient, a normal person, and you can see how many brain cells we had. Now, over time, in the Parkinson's disease infected cells, what we actually saw is we saw that those cells died. So again, this mimicked disease. So we had our disease in a dish. So I've just shown you 2 examples out of hundreds of why embryonic stem cell models have proven extremely valuable. However, why might ES cells not be the very best model for every neurodegenerative disease? Why are we looking elsewhere? For example, ES cells with certain genetic diseases are not easy to obtain for study, and as I told you, we can use virus to give ES cells those genes, but that's not exactly how it happens in nature, so that might not be the best way to model the disease. Also, not all diseases have a known gene cause. For example, most cases of Parkinson's disease and most cases of Lou Gehrig's disease, there's no family history. There's no known gene as of yet. And lastly, ES cells, unfortunately, are associated with some controversy. This is why we wanted to move into iPS cells, which you heard about earlier, to model neurodegenerative diseases. And in particular, in our laboratory, we're focusing on two diseases, spinal muscular atrophy and Huntington's disease. So I'm just going to give you a brief reminder after lunch about what are these iPS cells everyone is talking about. So I mentioned earlier, to get what the embryonic stem cells, you take a fertilized egg, then take these cells, and we can grow these cells in the lab and make any other cell type from them. Well, in 2008, two different groups, one in Japan and one in Wisconsin, actually found out that you can take skin cells from an adult human and induce pluripotency. Now, what do we mean by this? Induce, to bring about by influence, pluripotent, the ability to mature into many different cell types. Or to boil down this science jargon, basically what we're saying is we took skin cells and made them almost identical to embryonic stem cells. And we did this by giving a gene cocktail, if you will, to the skin cells that made them stem cells, nearly identical in every fashion and able to make almost any tissue type in the body. And what's really interesting is when you take these cells from a patient that has a neurodegenerative disease, you take the skin cells, give them this gene cocktail, then grow them into brain cells, you actually get brain cells that are diseased, as are in the patient from which you took the cells. Another way we like to think about this is what we call the Pinball Model. Now, this is actually the brainchild of one of my fellow postdoctoral students, Dr. Drew Serene. Normally, in development and in making ES cells, you fertilize an egg with a sperm, and then you're able to make what we know as embryonic stem cells, which by following these various paths can make any tissue type, so skin, bone, lung, liver, heart, brain. And as was mentioned earlier, we used to think this was a one-way path, that once you got down here, you were stuck down here. But those two researchers I mentioned earlier, they found the flippers. So what they were able to do is they were able to take these adult stem cell types and flip it all the way back where we could roll down the pathway again. So you can take skin or blood, use these flippers, or that gene cocktail I mentioned, and flip it all the way back, then roll down a different path to the brain. It's really an absolutely amazing process. So many neurodegenerative models are currently being studied using these iPS cells. And as I mentioned in particular, in our laboratory, we're studying spinal muscular atrophy and Huntington's disease. Spinal muscular atrophy, it's a disease characterized by severe paralysis in children, and it's due to the death of the cells in the spine that connect to the muscles. It's an absolutely terrible disease that affects children. This disease, when it was first characterized, to kind of tell you how these babies are, is it used to be called floppy baby syndrome. These poor children can never hold their heads up, they can never roll over, and most of these patients do not survive past childhood. It's a truly terrible disease, and what's scary about it is that it's a silent disease. So 1 in every 35 people is a silent carrier of this disease. And if 2 silent carriers have a child, they have a 1 in 4 chance of having this disease. So most parents of children with SMA, they don't have any family history. So what we were able to do is we actually got skin cells from a patient, and we gave them the gene cocktail to make these stem cells, these iPS cells. Then we were able to mature in the laboratory these stem cells into the spinal cells that die in the disease. You can see here they're shown in red. So we wanted to see, can we make a disease in a dish for spinal muscular atrophy? On the left, you can see those spinal cells. Now these cells were taken from the unaffected parent. If you remember, the parents are just silent carriers. They look like you and I. And from these, you can see on the right, these are the cells from an SMA patient. What ended up happening was that these spinal cells ended up dying in our culture dishes. And so we were able to achieve our goal of disease in a dish, and this was really exciting for us because this is actually this was the first report using these iPS cells of achieving the goal of disease in a dish. It was the first time it had ever been shown using this new type of stem cell, so it was really exciting to us. So we've shown that in a childhood disorder, we can use iPS cells to model disease. We wanted to look at a very devastating disease, as you just heard, Huntington's disease, and a more adult neurodegenerative disorder. As you heard, it's a progressive brain disorder that causes uncontrolled movement, emotional problems, and eventual loss of cognition. It's generally caused, you inherit a gene from either your mother or your father. And also, as you heard, usually by the time you're diagnosed, you have already had children. So Huntington's disease, you can see here on the left, the brain of a Huntington's disease patient as compared to a non-disease person. And what I'd like you to focus in is this red arrow. You can see right here, this region of the brain, in the middle of the brain, called the striatum, is the first part of the brain to die. And so those brain cells are the most susceptible to this disease. So in working with the consortium that was actually mentioned this morning, 8 labs across the globe, one actually here at UC-Irvine, Dr. Leslie Thompson's lab. We took samples actually from several Huntington's disease patients. We took their skin cells, used the gene cocktail, and made our stem cells. Then we were actually able to mature these stem cells into those very specific brain cells that are in the middle of the brain. They're called medium spiny neurons. They're very beautiful. But this is to bring up that, unfortunately, science is not always a simple story. I've just shown you 3 cases where we got our stem cells, we matured them into the cells that die, and boom, we had our disease in a dish. Well, unfortunately, in this story, that's exactly what we expected when we set out on this adventure. And then we all grew them out to about 100 days, and they were perfectly fine. So we didn't have our disease in a dish, which at first was extremely disheartening. But then we had to think of the big picture. So these cultures are 100 days old. Human disease, generally onset is somewhere around 45. Well, if you think about it, even with our most severe case where the poor child had onset at 6 years old, 100 days pales in comparison to that time frame, and unfortunately that's too long to keep cells in culture. And for other adult neurodegenerative diseases, as we heard this morning actually, it was a great point that they don't always see the same thing. So in some adult onset diseases, what we're finding is you have to stress the cells to see a phenotype. And we know even in our everyday lives, stress can age you faster. I mean, look at the president's hair. Obviously, some things are very stressful and can age you quite rapidly. So this is actually also showing promise for our Huntington's disease cells. Is we are using such things as toxins or taking away the goodies in their food, and this is really showing to stress out the cells. They don't like it, and we're hoping to see a phenotype. So we have our disease in a dish. Why? What are we going to do with them? As was mentioned earlier, the main goal in our lab is drug screenings. For many diseases, there aren't great therapies. Huntington's disease, there's really not much. Same with spinal muscular atrophy. So here you can see some cells. Basically, to sum up this drug screening that we did, we wanted to look for an increase. You'll see the arrows, these little green dots. And what we saw is after we gave them several compounds, we saw an increase in the dots, which was great. That's exactly what we wanted to see. So what we're trying to move now is into high throughput screening. Again, this is science jargon for we want to screen a lot of cells with a lot of compounds all at the same time and speed up this process. So here's a picture of a plate. This is a kind of an outdated picture. Now we have plates that hold over, they have over 1,000 compartments in one plate where you can actually put cells in these compartments and test thousands of drugs at once at various dosing, and then take pictures of the cells. And our goal is to see if we can find any compounds that help the cells to live. And a lot of this high throughput screening that's now going on in our lab is thanks to CIRM. So what do we envision is the future of disease modeling and with iPS cells? We feel that iPS cells, there's great potential for understanding the mechanisms of genetic disease and finding new drugs. There are many more disease models on the way, and high throughput screening or screening a lot of compounds at once is now crucial. I'd like to leave you with this guy. Now, this is a jellyfish, and what's actually marvelous about this jellyfish is it's its own iPS cell. This jellyfish lives very deep in the ocean and was recently discovered that when the jellyfish starts to get too old, it can turn on its own genes, move backwards through development to its very early embryonic polyp stage. Then it can hang out till the stress is gone, there's more food, and it can entirely regrow again. And there's not much that's been studied on this jellyfish because of where it lives, but from what we know, it can happen indefinitely. It's the fountain of youth, it's immortal, and it just goes to show as much as scientists think they're clever, Mother Nature's got us beat. So in conclusion, modeling neurological illness using human stem cells provides a novel way to study neuron diseases and discover new drugs, and we truly believe that iPS cells could be the future for disease modeling. So I'd like to thank you all for letting me speak to you today. I'd like to thank everyone at Cedar-Sinai, especially my boss, Clive Svensson, as well as our collaborators and the HD Consortium and our funding, and especially CIRM, and thank you all very much. Happy to answer any questions.
I was under the impression that we would be able to use stem cell research to get rid of the disease in the future if a person was identified, that there'd be a way to genetically change so the person would not have it. Is that possible?
Oh yes, the question was that she was under the impression that we could use stem cells to entirely get rid of the disease. From what I've heard, that might, I guess from what I've heard, that might not be possible. I know they're using, for some diseases, they're talking of using virus to change the genes. Also, a lot of the promise of stem cells would come with transplantation that you've heard of earlier that is still in its very, very baby stages. So using stem cell therapy to treat disease, to grow that part of the brain that would die in disease in a dish and then do transplants. So, but in terms of changing the all the genes in your body, I'm not quite certain about that. If anyone else had?
I might answer that in a slightly different way. So for a disease like Huntington's disease, where we know what the mutant gene is, and we know there's only one copy of it, that there is a way to potentially take skin cells from an individual with Huntington's disease, that's a difficult word to say, Huntington's disease, correct that gene mutation in those stem cells, and then turn them into a cell type that you can transplant back into the patient. And Leslie Thompson and I here at UCI are working on that, using a technology that was developed by a company up in San Francisco. That type of approach, though, is going to be more difficult for other so-called polygenic diseases where there may be lots of different mutations involved and we don't even know the gene at all. But for Huntington's disease, there potentially is a strategy to do that.
Thank you.
I know I've heard this numerous times, but today it finally clicked, that you said that the iPS cells are nearly identical to embryonic stem cells. In what ways are they different?
Sorry, the question is, I said nearly identical to embryonic stem cells, not completely identical. I'm trying to think of a way of phrasing this without going deep into science jargon. So they are, by all terms, they can make any cell type in the body. What's known is we're finding that with these early iPS cells, they do have a slight memory of the cell type from which they came. So they are truly a stem cell. They can make any type of cell in the body, but they remember that they were once a skin cell. But what's really interesting is just within the past couple of months, there have been researchers who have found a slightly different way of making these iPS cells where they're starting to lose that memory and becoming pretty much identical to embryonic stem cells. So it's more refining the process to make them exactly like embryonic stem cells. But that was a great question. Did it answer your question? Okay, great. In the back? How long do these cells live in culture? That's a great question. The longest, it really depends upon your cell type of interest. Certain cells live longer in culture than others. For Huntington's disease, these brain cells of the middle brain, the longest I personally have gotten them to live in culture is about 130 days. After that, it's a very stressful environment, and basically the way we grow them isn't as good, but we're learning every day how to keep these cultures growing for longer and longer. It's kind of like a dance and an art to try to get these cells to grow for a very long time in culture.
What happens to them?
So to grow them, they stick to the bottom of a plate. So it's more of a technical problem of after a certain time, the plate, the sticky substance that's on the bottom, the sticky substance just deteriorates. And so the cells float up, and the cells like to be stuck to something. So we're trying to develop better sticky material to keep them stuck to the bottom of the plate. So it's not so much that they die, it's that the conditions just aren't supporting them anymore. Okay, great. Thank you.